Hi guys, thank you for joining me today. I'm going to do a lesson on transport in plant and photosynthesis. So if you have been struggling with the year 12 content and how it links to the year 13 content, then why not check out this video? Hope you like it and you find it helpful. Do ask questions if you're not sure at any point. The first thing I want to look at are plant cells. So we know that plant cells are eukaryotic, meaning that they contain linear DNA associated with histone proteins, have membrane-bound organelles and ATS ribosomes. However, they also have additional features that distinguish them from other eukaryotic cells such as animals, fungi and protists. So what are these features? Well, one of the most important identifying structures that gives the plant cell its shape is the cell wall. The cytosol, the liquid medium inside the cell, is where many chemical reactions take place. Now, water moves into and out of the cells by osmosis from a higher water potential to a lower water potential across the partially permeable membrane. So to prevent the cell from bursting and to help maintain structural rigidity, plant cells are surrounded by a cell wall made of cellulose. Now, this provides strength and support whilst allowing water and dissolved substances to pass through. So what is cellulose then? Well, cellulose is a polysaccharide made from many beta-glucose molecules. Remember, to identify a beta-glucose molecule, you need to look at carbon number 1. The hydrogen is below and the hydroxyl group is above. So the polysaccharide cellulose is made from many beta-glucose monomers held together by glycosidic bonds. So to allow a condensation reaction to occur between the hydroxyl group on carbon 1 of one glucose molecule and carbon 4 of the next glucose molecule, every alternate beta-glucose molecule must rotate 180 degrees. Now this rotation ensures that the hydroxyl groups are aligned correctly and results in the formation of a straight, unbranched chain polymer. So that is what we call a cellulose molecule. So the cellulose molecule is this long chain of the beta glucose held together by a 1-to-4 glycosidic bond. So we have the fact that these parallel cellulose chains lie close together. And because they lie close together, they form what's known as crosslinks by hydrogen bonds. Now, these crosslinks produce these strong microfibrils, and these hydrogen bonds prevent access by water molecules, making cellulose highly resistant to hydrolysis. As a result, the cellulose is an excellent structural molecule, providing strength and rigidity to the plant cell walls. So if we look at the cellulose fibres then, cellulose fibres are organised into bundles of microfibrils which group together to form macrofibrils. These create a strong but porous mesh in the cell wall that is freely permeable to water and most dissolved substances. In contrast, the plasma membrane, which lies underneath the cell wall, forms a selectively permeable barrier between the cytoplasm and the external environment. Now, it contains transport proteins and therefore regulates the solute movement, helping maintain cellular homeostasis. So, we know that plant cells are connected by plasmodesmata, these tiny little channels crossing the cell walls. Now, each plasmodesmata is lined with the plasma membrane and contains a thin strand of endoplasmic reticulum, known as the desmotubule. Now, these channels allow ions, small molecules, hormones, sugars, and even larger substances such as RNA and proteins to pass directly between the cells. This regulated cytoplasm to cytoplasm transport is called the symplast pathway. Now, plasmodesmata act like corridors with doors where proteins control what passes through. By contrast, the apoplast pathway involves movement through the cell wall and intercellular space, which are unregulated. Now, this pathway acts like a free-flowing highway, allowing rapid bulk flow of water and solutes between large numbers of cells. So if we look then at our roots and we start with the root hair cells. We know the roots are located at the base of the plant. We know they absorb water and mineral ions from the soil. And we know at the root tips, the cells of the apical meristem divide by mitosis and differentiate into these root hair cells. Now, these differentiated cells develop long, thin extensions that are going to greatly increase the surface area for absorption. Their thin cell wall provides a short diffusion distance, further enhancing this uptake. Now, substances can then move through the root cortex by two main pathways. The apoplast pathway we just discussed through the cell wall and intercellular space, unregulated, or the symplast pathway through the cytoplasm via the plasmodesmata, regulated. Now, to enter the symplast pathway, solutes must first cross the plasma membrane. Because mineral ions are charged and hydrophilic, they cannot diffuse directly through the phospholipid bilayer. Instead, they are moved into the root hair cells via active transport through carrier proteins, and it's against their concentration gradient. This accumulation of mineral ions in the cytoplasm increases the solute concentration and therefore lowers the water potential inside the cell. As a result, water from the soil enters the root hair cell by osmosis down the water potential gradient. So we can see here that we've got the mineral ions being actively transported against a concentration gradient. We are going to therefore lower the water potential inside that root hair cell, and the water will move by osmosis. Now, when the root hair cells have differentiated, they have lots of mitochondria, and that is to provide the necessary ATP for the active transport. So mitochondria is where we have respiration, aerobic respiration occurring, and because it's aerobic respiration, we have lots of ATP molecules. So this is where we could interlink our knowledge for respiration and apply it to how the roots are able to take in the water. Water can move through the root in two main pathways before it enters the xylem. The symplast pathway, water moves cell to cell through the cytoplasm connected by the plasmodesmata, or the apoplast pathway, where water moves through the cell walls and intercellular space without crossing a plasma membrane. Now, when water reaches the endodermis, the layer surrounding the vascular bundle, it encounters the Casparian strip. This strip is made up of suberin, a waterproof substance which blocks the apoplast pathway. As a result, all water and the dissolved minerals is forced to cross a plasma membrane and move into the symplast pathway before entering the xylem. This ensures that the plant can control what substances pass into the vascular tissue. Let's actually look at the xylem. Let's start with what the vascular tissue is made up of. And the xylem vessels are made up of dead, hollow cells that join end to end to form continuous tubes. Now, this allows water to move upwards through the plant. Meristem cells in the vascular cambium will divide by mitosis to produce living precursor cells, which then differentiate into xylem cells. Now, during differentiation, a primary cell wall made of cellulose is laid down. A secondary cell wall made of lignin, which is a complex irregular polymer, is deposited in specific patterns. Now, these patterns can be spiral, they can be ring-shaped, they can be net-like, or pitted. Now, this lignification strengthens the wall and prevents collapse under the tension created during water transport. After the secondary wall is complete, the xylem undergoes programmed cell death. So the xylem cell is actually activated to release hydrolytic enzymes which break down the nucleus, the organelles, and cytoplasmic macromolecules. This controlled self-destruction leaves behind a lignified hollow tube, allowing water to flow as one continuous column, and bordered pits that allow lateral water movement between adjacent vessels and surrounding cells. Now, lignin is hydrophobic, so it does not form hydrogen bonds with water. However, within the xylem vessel walls, there are pits and microsurfaces where cellulose remains exposed. Now, cellulose has got a part of it which is hydrophilic because it contains hydroxyl groups that can form hydrogen bonds with water molecules. Now, the interaction between water molecules and the cellulose in the vessel wall is known as adhesion. And adhesion helps water remain in contact with the xylem wall and remain in the upwards movement of water through the plant by capillary action. The narrow diameter of the xylem vessel further enhances this capillary action, supporting the continuous water transport from root to leaves. So, if we're wanting to visualize this, to observe how water and mineral ions are distributed up the xylem vessel, we can do this by placing plant stems in water containing a strongly colored dye. The dye will be carried along with the water up the xylem vessel, staining the walls. After 24 hours, you can take a transverse and longitudinal cut through the plant stem using a very sharp blade, and what will happen is the dye will show the position of the xylem vessel as either a coloured region, which is visible in coloured dots, the transverse section, or coloured lines like this diagram. Now, what you can also do is you can take a stem and you can show that different xylem strands transport water and mineral ions to different parts of the plant by splitting the base of the plant stem lengthwise into two sections while leaving the upper part intact. Now, each section is placed into a separate test tube, one containing plain water and the other containing the coloured dye. The dye will travel up the xylem in that section of the stem, colouring only the regions of the flower which are supplied by those xylem pathways. And that is shown in my diagram here, where I had a blue dye. One side became coloured with the blue dye, whereas the other side did not because it was only supplied with water. Let's talk about the phloem. Now, the phloem is an essential tissue in plants for transporting organic compounds such as sucrose and amino acids. Unlike water transport in the xylem, which moves upwards from roots to leaves, the phloem transport is bidirectional, moving substances both up and down the plant. Phloem sieve tube elements are produced when meristematic cells in the cambium divide by mitosis, producing daughter cells that are pushed outwards towards the periphery of the stem, or the root. Now, during differentiation, the daughter cells elongate into tube-like shapes. The end walls become perforated, forming sieve plates with many pores to allow the cytoplasm and dissolved solutes to flow between the cells. Most organelles, including the nucleus, are degraded, meaning the sieve tube element cannot function independently. Each sieve tube element is supported, therefore, by a neighbouring companion cell, which controls protein synthesis and provides ATP and metabolites through the plasmodesmata. The sieve tube elements align end to end via sieve plates to form continuous channels, and this allows for the mass flow of sucrose and amino acids. So we know that plants are multicellular organisms and therefore require an efficient transport system. So to move substances over long distances, as their surface area to volume ratio is too low for diffusion alone to meet their metabolic needs, the xylem and the phloem tissue are arranged together in these structures known as the vascular bundles. Now, these vascular bundles form a continuous network running from the roots through the stem and into the leaves, creating the plant's transport system for water, mineral ions, and assimilates such as sucrose and amino acids. Now, the arrangement of the vascular system varies in different parts of the plant, reflecting the specific roles of each organ. Now, the root, what we have here is that the primary xylem forms a star shape at the center of the vascular cylinder, usually with 3 or 4 arms. Now, the phloem is located between the regions of the xylem tissue. The central xylem provides mechanical strength, allowing the root to resist pulling forces as he anchors the plant into the soil. And the phloem transports organic nutrients to and from the root cells, supporting growth and storage. Now, as we move up the plant, we move on to the stem. Now, the vascular bundle in the stem are arranged in a ring near the outer edge of the stem. The xylem lies towards the inside and the phloem towards the outside, and a layer of cambium, a meristem, sits between. Now remember, the cambium, being meristem, is full of undifferentiated cells that have the capability of going through mitosis, dividing, producing daughter cells, and then those daughter cells differentiate into either the xylem or the phloem. Now, the arrangement of the xylem and the phloem actually provides the strength and flexibility which helps with the stem resisting the bending force and remaining upright. After the stem, what we have is the leaf. So the vascular bundle forms the veins of the leaf. Within each bundle, the xylem is actually on the upper side, transporting water for photosynthesis and maintaining turgor pressure. The phloem lies beneath the xylem and is responsible for the translocation of those sucrose from the source, the leaves, to the sink, the roots of the fruits. To understand how water moves through the plant from the roots to the leaves, we must consider the structure and property of water. A water molecule is made from two hydrogen atoms covalently bonded with one oxygen atom. And in a covalent bond, each atom nuclei attracts the shared pair of electrons, and the attraction is called electronegativity. If the electronegativity difference is large, one bonded atom will have a much greater attraction for the shared pair of electrons than the other bonded atom. Now, the oxygen atom in a water molecule is more electronegative than hydrogen and therefore attracts the shared electrons more strongly. This gives the oxygen atom a partial negative charge and each hydrogen atom a partial positive charge, making water a polar molecule. Now, due to this polarity, when two water molecules are close together, hydrogen bonds will form between them. These weak intermolecular forces occur between a hydrogen atom covalently bonded to an electrical negative atom such as oxygen, nitrogen, or fluorine in one molecule, and a lone pair of electrons on an electronegative atom such as oxygen, nitrogen, or fluorine in another molecule. So in water, a hydrogen bond forms specifically between the hydrogen atom of one molecule and a lone pair of electrons on the oxygen atom of another water molecule. This intermolecular bonding gives water its cohesive property. Because many hydrogen bonds form between water molecules, a large amount of energy is required to overcome them. So at room temperature, the molecules do not have enough kinetic energy to completely break these hydrogen bonds, so water remains liquid rather than becoming a gas. This means that water molecules can move freely past one another while still being held together by temporary hydrogen bonds. So water is often called the universal solvent because its polarity allows it to dissolve many ionic and polar substances. If a molecule is polar and contains an electronegative atom, such as oxygen, nitrogen, or fluorine, it can form hydrogen bonds with water molecules. Now, these interactions occur when the electronegative atom in the solute attracts the partial positive hydrogen atoms in water. Such regions are described as hydrophilic. So we have these water-loving regions because they readily attract and interact with and dissolve in water. The polarity of water also allows it to interact with ions surrounding them. So the delta negative oxygen atoms of water will surround cations, while the delta positive hydrogen atoms are attracted to anions, and what they form is what's known as a hydration shell. So this stabilizes the ions in solution and explains why many salts will dissolve readily in water. In addition to its interaction with ions and polar molecules, water also exhibits adhesion. Now, this is the ability to form hydrogen bonds with other polar or charged surfaces. So we spoke about that when we talked about the xylem vessel. While cohesion describes the attraction between the water molecules, adhesion occurs when water molecules are attracted to a different material, such as the walls of the plant xylem or glass. Now, these adhesive interactions combined with cohesion allow water to climb narrow tubes against gravity. That is a process known as the capillary action, and it plays a vital role in transporting water in plants. So to understand the driving force of water up the plant from the root to the leaves, we need to consider another property of water. That is the latent heat of vaporization. So water molecules form hydrogen bonds with one another, which is why water remains liquid at room temperature. When water changes state from a liquid to a gas, water vapor, energy is needed to break the hydrogen bonds. The amount of energy needed is high, and this is referred to as the latent heat of vaporization. Individual molecules at the surface that gain enough kinetic energy can overcome these intermolecular forces and escape as water vapor. When a water molecule evaporates from the mesophyll cell surface into the airspace, cohesive forces between water molecules pulls more water into its place, while adhesive forces between water and the cellulose cell wall help maintain a continuous column of water from the xylem to the site of evaporation. This creates tension that draws water upwards through the plant. As plants rely on water for many essential processes, including chemical reactions, maintaining turgidity, and photolysis during photosynthesis, it is important that they maintain a constant supply of water. Therefore, the structure of the leaf is specifically adapted to reduce excessive water loss while still allowing gas exchange for photosynthesis and respiration. The waxy cuticle covering the lower and upper epidermis acts as a waterproof barrier, reducing evaporation from the leaf surface. The palisade cells near the upper surface of the leaf are compacted together. This maximizes the absorption of light for photosynthesis whilst also reducing the exposed surface area for evaporation to occur. The lower surface of the leaf contains small pores known as stomata, and the stomata are surrounded by guard cells, specialized epidermal cells that control their opening and closing. During differentiation, the guard cells develop several unique structural and functional adaptations that enable them to carry out the role effectively. They have the ability to respond to stimuli. The guard cells are really sensitive to light, carbon dioxide concentration, and water availability, enabling the stomata to open during the day for gas exchange and photosynthesis and close at night or under water stress to reduce water loss. They also have an uneven thickening of the cell wall. The inner cell wall of the guard cell is thicker and less elastic, while the outer cell wall is thinner and more flexible. So plants need to photosynthesize. Therefore, they require a supply of water for that light-dependent reaction and carbon dioxide for the light-independent reaction. When light levels for photosynthesis are detected by guard cells, they respond by opening the stomata. The guard cells actively pump hydrogen ions out of the cell. This causes the membrane to become hyperpolarized, more negative. This results in voltage-gated potassium channels opening and potassium ions then diffusing into the guard cells by facilitated diffusion. This increases the solute concentration inside the cell. As a result, the water potential decreases and water enters by osmosis, causing the cells to become turgid. As the inner wall of each guard cell is thicker and less elastic than the outer wall, turgor pressure causes the cells to bend outwards, opening the stomatal pore. In low light conditions or in response to the hormone abscisic acid, ABA, the guard cells actively transport potassium ions out of the cell. This reduces the solute concentration and raises the water potential inside the cells. Consequently, water leaves by osmosis, causing them to become flaccid, and this loss of turgor pressure results in the stomatal pore closing, preventing further water loss. So when water molecules evaporate from the mesophyll cell surface into the air space, this creates a concentration gradient. When the stomata are open, the water vapor will diffuse down the concentration gradient out of the leaf. This loss of water vapor from the plant is called transpiration. So transpiration is the evaporation and diffusion of water vapor out of the plant through the leaves or the stem. Now we can estimate the rate of transpiration using a potometer. A potometer measures the water uptake by a plant by measuring the distance moved by an air bubble along a scale over a specified time. A leafy shoot is cut underwater and at a slant to increase the surface area for water uptake and to prevent air from entering the xylem. This avoids the formation of air bubbles, which would break the continuous column of water by disrupting the hydrogen bonds between the water molecules and stopping the transpiration pull. The shoot is then inserted, also underwater, into a capillary tube of a known diameter, for example, 1 millimeter. Ensuring that the apparatus is watertight and airtight, you can put Vaseline around the area. Other end of the capillary tube, an air bubble is introduced and its starting position on a calibrated scale is recorded. After a specified time period, for example, could be 30 seconds, could be a minute, could be an hour, the distance moved by the air bubble is recorded. This can then be used to calculate the rate of transpiration in millimeters cubed per minute. Now first you will need to calculate the volume of the capillary tube using the formula pi r squared length. So if we have our diameter, which was 1 millimeter, we would therefore work out our volume of the capillary tube by doing pi times the radius 0.5 squared times the distance that it moved. Now in 30 seconds it moved 18 millimeters. So pi times 0.5 squared times 18 would give us an answer of 14.71. We would then need to multiply that number by 2 to calculate the rate in 1 minute. 14.71 times 2, 29.42. Now, transpiration occurs through the stomata. So when using the potometer, the total leaf surface area should be considered, as well as the number of leaves present. To estimate your leaf surface area, each leaf can be removed and flattened onto graph paper. The total area is then determined by counting the fully covered squares and adding estimates for partially covered squares. Once you have figured that out, remember you must multiply by 2 because there are 2 sides to the leaf. We calculate the rate of transpiration to be 29.42 millimeters cubed per minute. If we estimate the total leaf area of the shoot to be 150 centimeters squared, we can then work out the rate of transpiration. We could calculate the rate of transpiration in centimeters cubed per hour per centimeter squared. As transpiration is due to the evaporation and diffusion of water vapor from the leaf, the rate of transpiration can be affected by different environmental factors such as light intensity. High light intensity causes stomata to open wider, increasing the rate of transpiration. However, it can have a knock-on effect to the closing of stomata. Temperature, faster evaporation from the mesophyll cells, faster rate of diffusion of water vapor out of the leaf. Warm air holds more water vapor, resulting in a steeper gradient. humidity. Now, humidity is the concentration of water vapor in the air. So if we have low humidity, it will mean we've got a steeper gradient and therefore faster transpiration. If we have high humidity, we would reduce our concentration gradient, therefore slower diffusion, less transpiration. So if we were to place a bag over our leafy shoot, we would create a humid environment, and that would therefore be a way in which we could see how humidity has an effect on the rate of transpiration. Now, wind, i.e. air movement, is going to maintain a steep gradient and therefore increase the transpiration. So again, if we were to plan an investigation on this, we could use a fan with differing speeds so that we can see how that air movement affects the rate of transpiration. Now, water availability is obviously going to have an effect on the rate of transpiration. If there is low soil water, that is going to result in stomatal closing and therefore less transpiration. So the stomatal density can also affect the rate of transpiration. It can be measured by applying clear nail polish to a leaf surface and allowing it to dry to form an impression. The dry film then can be peeled off and placed on a microscope slide, and the number of stomata in several fields of view can then be counted. The field of view area is determined using an eyepiece graticule and stomata density. So stomata per millimeter squared is calculated. So if I have the diameter of my field of view being 1.7 millimeters, I can calculate what my radius is, and then I can use that to work out the area of a circle, pi r squared. So if I have my diameter of 1.7 and I divide that by 2, I would therefore get a radius of 0.85. My area of my circle, I would then substitute the numbers in, so pi times 0.85 squared would give me the area of the circle being 2.27. Now, if I count the number of stomata that we can see here open in my field of view, there are 17 stomata. So now I would just divide that into an equation. So the number of stomata is 17 divided by the area of field of view, which was 2.27. That tells me that my stomatal density is 7.49 stomata per millimeter squared. Now, excessive water loss through transpiration can be problematic in environments when water is scarce. So plants have adapted to reduce water loss, such as having a waxy cuticle, the stomatal positioning, and extensive root systems to maximize water uptake. Plants inhabit a wide range of environments, from woodlands, meadows, sand dunes, and streams, with varying availabilities of water. Therefore, plant adaptations differ according to their habitats. Now, xerophytes, i.e. cactus, marram grass, aloe vera plants, live in very dry environments, and they show adaptations that include things like a reduced surface area. They have small leaves, or they roll their leaves, or their leaves have turned into spines. They have thicker waxy cuticle on both the upper and the lower epidermis. The stomatal adaptation is there is a reduced density of stomata. The stomata can be sunken. Now that's going to reduce the air movement and create a humid environment close to the stomata. They can also have hairs surrounding the stomata, again trapping a layer of air, making the concentration gradient less. Or it may be that they only open the stomata at night, i.e. in cooler temperatures, therefore there's less transpiration. The stomata opening at night is a way in which they can bring in the carbon dioxide. However, they need to have a way of storing that so that they can then use it during the daytime when there is light available. And the other thing that a xerophyte will do is have a place where it can store the water. So water storage within tissue means that it can store it when it is scarce. Now, the other type of plants that you need to know about are known as hydrophytes. So these are examples like water lilies. They live in aquatic environments and their adaptations are different. They have very thin waxy cuticles. They have many open stomata, but the stomata that they have are on the upper surface, allowing for that gas exchange to occur with the air rather than on the lower surface, because the lower surface is submerged in water where there is less oxygen dissolved. They have wide, flat leaves so they can absorb light for photosynthesis, and they have these air sacs that allow for buoyancy, so keeping them upright so they can maximize the amount of light that they are absorbing and allow for that gas transport. Photosynthetic tissue contains cells specialized to carry out photosynthesis. Palisade mesophyll cells are highly adapted to maximize the rate of photosynthesis. They have many chloroplasts to absorb large amounts of light. They are elongated, so they tightly pack together to maximize the amount of light they're capturing. They have thin cell walls to allow rapid diffusion of carbon dioxide, and they have a large central vacuole to maintain the turgor pressure and keep the cells closely packed. Chloroplasts are the site of photosynthesis in plant cells. The actin-based cytoskeleton helps reposition the chloroplast to maximize light absorption. Photosynthesis uses water and carbon dioxide to produce glucose and oxygen. The glucose acts as a respiratory substrate and the oxygen is required for aerobic respiration. Meanwhile, respiration releases carbon dioxide and water, which can be used again in photosynthesis. This establishes an interdependent relationship between the two processes. The structure of the chloroplast is important for photosynthesis. Evidence for its evolutionary origin includes its double membrane, circular DNA, and 70S ribosomes. These features support the endosymbiotic theory, which proposes that chloroplasts evolved when a photosynthetic prokaryote was engulfed by a larger cell and formed a mutually beneficial symbiotic relationship. Thylakoids are flattened sacs consisting of thylakoid membranes surrounding the thylakoid space. They are stacked to form grana, which are connected by membranous channels called lamellae. These membranes provide a large surface area for the absorption of light because many photosystems, electron carriers, and ATP synthase are embedded in the thylakoid membrane. Their flattened shape creates a small internal space, the thylakoid lumen, enabling the accumulation of hydrogen ions and the formation of a steep proton gradient. So to understand how chloroplasts absorb light, we need to take a step back and we need to remind ourselves about visible light. So visible light from the sun is composed of a range of wavelengths, 400 to 700 nanometers. Now the wavelength is the distance from one point of a wave to the same position on the next wave. For example, peak to peak, or it could be trough to trough. When we see a rainbow, we observe all the colours in visible light: red, orange, yellow, green, blue, indigo, and violet. This occurs because white light entering water droplets in the air is refracted, internally reflected, and dispersed, causing different wavelengths to spread out at different angles and form the spectrum of colours that we see. So photosynthetic pigments then absorb these different wavelengths of light and are embedded in the thylakoid membrane within protein complexes to form what's known as a light harvesting system, the antennae complex. The primary pigments, chlorophyll A, absorb mainly red and blue wavelengths of light and reflect green light, which is why most leaves appear green. Accessory pigments, including carotenoids, xanthophyll, and chlorophyll B, absorb additional wavelengths of light and pass the energy to chlorophyll A. This broadens the range of light that can be absorbed, maximising the efficiency of photosynthesis. In autumn, chlorophyll production decreases and breaks down more rapidly. As its green colour fades, the colours of the accessory pigments become visible, making leaves appear green, then yellow, then orange, or even red. Photosynthetic pigments can be extracted from leaves and then separated using thin layer chromatography. A TLC plate has a thin coating of silica gel, an adhesive substance which acts as the stationary phase. A pencil line is drawn approximately 1 centimetre from the bottom of the plate, and then the pigment extract is then applied to the line, 1 drop at a time, allowing each to dry before adding the next one. A darker initial spot indicates a higher concentration of pigment. The TLC plate is placed upright in a beaker containing an organic solvent, the mobile phase. As the solvent moves up the plate by capillary action, the pigments dissolve and travel with it. Each pigment has a different solubility in the solvent and different attractions to the stationary phase. This causes them to move at different speeds, resulting in separation into distinct bands on the plate. Now, we can then work out the retention value for each pigment by using the distance moved by the pigment divided by the distance moved by the solvent, and we call that the solvent front. So if you are given in your exam a diagram that shows a picture like this, you would take your ruler, you would measure from the origin to the solvent front, and you would measure from the origin to whichever pigment they want you to observe. That will therefore allow you to calculate the RF value. Now, it may be that you calculate the RF value for a green pigment to be 0.64 and then conclude that it was probably chlorophyll A, and you can tell that because it shows a bluey green colour, which is what you would expect. However, if you're given a table of results that say that chlorophyll A is 0.63 and you calculated a value that wasn't that was named on the table, then that would be evidence against the conclusion. So be aware that expecting you to know how to calculate your RF value, but also how to then use that to draw a conclusion. Photosynthesis can be divided into 2 stages. The light-dependent stage. Light energy is absorbed by photosynthetic pigments and converted into chemical energy, forming ATP and reduced NADP. Light-independent stage, also known as the Calvin cycle. The products of the light-dependent reaction, ATP and reduced NADP, are used to fix carbon dioxide and synthesize organic molecules such as glucose. So to understand how this happens, we first need to look inside the thylakoid membrane. Inside the thylakoid membrane, photosystems are embedded, consisting of these antennae complexes and a reaction center. 2 types of photosystems, each with its own reaction center. So there is photosystem 2, PS2, and photosystem 1, PS1. The antennae complex captures light energy and transfers it to the reaction center. When a pigment absorbs a photon of light, its energy is transferred to one of the pigment's electrons. This causes the electron to gain energy and move to a higher energy level, the excited state. The energy can then be passed from one pigment molecule to another until it reaches the reaction center. Once at the reaction center, the chlorophyll A molecules can donate their excited electrons to the electron transport chain, initiating the process leading to ATP and reduced NADP formation. So a photon of light gets absorbed, excited electrons move to a higher energy level, energy is released as those electrons return down to their original energy state. So what's actually happening is quite complex, but let's see if we can figure this out. The photon of light can be absorbed by both photosystems simultaneously, allowing each reaction center to donate excited electrons to the electron transport chain. If NADP is available as the final electron acceptor, then non-cyclic photophosphorylation can occur. Electrons from photosystem 2 travel through the electron transport chain, passing through a proton pump to photosystem 1. Electrons from photosystem 1 are transferred to NADP reductase, which reduces NADP to reduced NADP. This movement of the electrons through the proton pump releases energy, which is used to actively transport hydrogen ions across the thylakoid membrane, creating a proton gradient. These hydrogen ions then diffuse back through ATP synthase, also embedded in the thylakoid membrane, driving the production of ATP. Now, to replace the electrons that were lost from photosystem 2 that went to photosystem 1, photolysis of water occurs. Water molecules are split into electrons, hydrogen ions, and oxygen, which is released as a waste product. If NADP is not available to accept electrons, cyclic photophosphorylation occurs in this process. Electrons from photosystem 1 are recycled, moving through the electron transport chain and across the proton pump back to photosystem 1. The electron flow maintains the proton gradient across the thylakoid membrane, allowing ATP production to continue even without the formation of reduced NADP. So it's important to understand the electron transport chain is required to pump hydrogens against the concentration gradient so that they then accumulate within the thylakoid space. This then results in a electrochemical gradient, and those hydrogen ions will flow down the electrical chemical gradient through the ATP synthase. That is chemiosmosis. So once the light-dependent reaction has occurred, we can either end up with ATP molecules, or if it's non-cyclic photophosphorylation, it's ATP and reduced NADP. The light-independent reaction, also known as the Calvin cycle, takes place in the stroma of the chloroplast. It does not directly require light, however, it depends on the products of the light-dependent reaction, ATP and reduced NADP. A molecule of carbon dioxide is fixed to a 5-carbon compound, ribulose bisphosphate, RUBP, forming an unstable 6-carbon intermediate molecule. This reaction is catalyzed by the enzyme RUBP carboxylase, commonly known as Rubisco. Due to the instability of the 6-carbon intermediate, it immediately splits into 2 3-carbon molecules of glycerate 3-phosphate, also known as GP. Now, each GP molecule is converted into a triose phosphate. This conversion requires 1 molecule of ATP and 1 molecule of reduced NADP. Now, during the process, reduced NADP is oxidized, therefore regenerating it so it can return to the light-dependent reaction to accept further electrons and hydrogen ions. The whole reduction stage requires ATP and reduced NADP, which were produced in the light-dependent reaction. So products from the light-dependent reaction are required for the reduction process in the light-independent reaction. Now, the RUBP used during carbon dioxide fixation must be regenerated as well to allow the light-independent reaction to continue. Now, to achieve this, 5 molecules of triose phosphate are converted back into RUBP. This conversion requires energy which is provided by the remaining ATP molecules. 1 triose phosphate molecule leaves the cycle and is used to synthesize other carbon-containing compounds required by the plant. Now, how do we know this? Well, this was demonstrated by Calvin and his co-workers, who illuminated a suspension of green algae in the presence of radioactive carbon dioxide. Samples were taken at different time intervals after the addition of radioactive carbon dioxide. Chromatography was then used to separate and identify the carbon compounds formed. After 1 second, only glycerate 3-phosphate, GP, contained the radioactive carbon. After 5 seconds, additional molecules were found containing radioactive carbon, including TP, sugar phosphates, and RUBP. And after 30 seconds, the radioactive carbon was detected in many other molecules, such as sucrose, amino acids, and lipids. Now, the rate of photosynthesis can be measured in several different ways. We can measure the volume of oxygen produced during photolysis in the light-dependent reaction. We can measure the volume of carbon dioxide taken up, indicating the carbon fixation in the light-independent reaction. Or we can measure it indirectly by measuring the increase in biomass resulting from the conversion of triose phosphates into stored organic compounds. Now, environmental factors such as light intensity, temperature, and carbon dioxide can affect the rate of photosynthesis. If any one of these factors is in short supply, it will then limit the rate of photosynthesis, and this is known as a limiting factor. Now, we can carry out investigations to look at these limiting factors on the rate of photosynthesis. Now, one way in which we can do that is by limiting the light intensity. Light is needed as an energy source in the light-dependent stage. If light intensity is low, less ATP and reduced NADP are produced, therefore less GP will be converted to TP in the light-independent reaction. At very high light intensities, photoinhibition may occur, and this occurs when the electron transport chain becomes overloaded, leading to the formation of reactive organic species, ROSs, which damage chlorophyll molecules, can damage photosystem 2, and can damage the thylakoid membrane. So light intensity is a limiting factor. If it's too low, what we can see is that the light intensity is limiting the rate of photosynthesis, and then it increases until a point at which it plateaus. When it plateaus, then something else is limiting the rate of photosynthesis. So as you can see from my graph, we have different things that can limit the rate of photosynthesis: carbon dioxide concentration, temperature, or some other factor like the chlorophyll molecules, the photosystems, and the thylakoid membrane, which need to be repaired. Now, if we look at temperature, we know that photosynthesis is a set of enzyme-controlled reactions. If we have low temperatures, we have less kinetic energy, less successful collisions, less enzyme-substrate complexes, therefore a lower rate of reaction. Now, if we increase temperature too high, that can result in more kinetic energy, more successful collisions, a higher rate of reaction until enzymes such as rubisco can become denatured. Now, carbon dioxide concentration is a really interesting one. When carbon dioxide levels are very low and or the temperature is above 25 degrees C, oxygen can bind to rubisco. Oxygen, therefore, is a competitive inhibitor for rubisco. Now, this leads to the production of a toxic substance that must be removed from the plant. Energy from ATP is required to convert this toxic substance into a useful metabolite, such as glycerate, which can then re-enter the Calvin cycle. And this process is known as photorespiration. Now, we can measure the rate of photosynthesis using a pH indicator because carbon dioxide affects the acidity of water. We know that carbon dioxide dissolves in water to form carbonic acid. Therefore, if we were to place a water plant, for example, like Elodea, or make algal balls, which you can see in my diagram, we have got these in a solution containing a pH indicator. Now, carbon dioxide with water reacts to form carbonic acid. Carbonic acid can dissociate into hydrogen ions and hydrogen carbonate ions. So if there is more carbon dioxide, we would expect there to be a dissociation of more hydrogen ions, therefore lowering the pH, making it more acidic. If there's less CO2 in the solution, we would have fewer hydrogen ions, therefore a higher pH. We would expect there to be a more alkaline solution. So if we observe the colour change over time after we have exposed these algal balls or water plants to specific intensity of light, we should see a change in the colour. Now, if it has gone a purple-red colour, that indicates that carbon dioxide has been uptaken by the organism and therefore photosynthesis is occurring. If the colour is a yellow colour, that is indicating there is a high carbon dioxide concentration in the solution, therefore photosynthesis is slow or absent. Now, this method is an indirect way of measuring carbon dioxide uptake. It's not the exact rate. For more precise measurements, we would want to use a CO2 sensor, and that would give us an actual reading of the carbon dioxide concentration. So the next thing we need to think about is photosynthesis uses water and carbon dioxide to produce glucose and oxygen. The glucose acts as a respiratory substrate, while the oxygen is required for aerobic respiration. Not all parts of a plant can photosynthesize because not all parts have chloroplasts that enable them to be able to do the process of photosynthesis. Therefore, molecules must be transported from the site of production, known as the source, to non-photosynthesizing cells, known as the sinks. So to allow transport, triose phosphate produced during the light-independent reaction is converted to assimilates, such as sucrose, amino acids, lipids. Now, we're going to focus on sucrose. So the sucrose, which is produced through the glucose and fructose phosphate being combined to form a disaccharide, can be loaded into the phloem via one of two pathways. So the first pathway could be the Symplast pathway. Remember, the Symplast pathway is where whatever the substance is, whether it be water or in this case sucrose, is going to move between the cells through those plasmodesmata, the cytoplasmic connections, eventually entering the phloem sieve tube element. Now, other way in which we can load sucrose is known as the apoplast pathway. Now, the plasma membrane of source cells contain sucrose transporters that move sucrose out of the cytoplasm into the cell wall and intercellular space. Now, the sucrose will then move towards the companion cell where it will be transported back into the cytoplasm. To understand how that is transported back into the companion cell, we need to understand the active transport of hydrogen ions. So at the companion cell, hydrogen ions are actively pumped out of the companion cell into the cell wall using ATP. This establishes a steep hydrogen ion concentration gradient. As the hydrogen ions diffuse back into the companion cell down their gradient, they pass through another co-transporter protein which simultaneously transports sucrose into the companion cell. So that is the apoplast pathway. Now, after the sucrose has been loaded into the companion cell, it moves into the phloem sieve tube element via the Symplast pathway. Now, the loading into the phloem lowers the water potential inside the phloem, causing water to move from surrounding cells and from the xylem vessel by osmosis. Remember, the xylem vessel has pits which allow the lateral flow of water. So the water will move from the xylem into the phloem down the water potential gradient by osmosis. Now, the rigidity and incompressibility of water, along with the structure of the cell walls in the phloem, results in a buildup of hydrostatic pressure. Water will flow from a region of high hydrostatic pressure to regions of lower hydrostatic pressure. This mass flow can be bidirectional, moving up towards developing leaves or down towards the roots. At the sink, sucrose is unloaded from the phloem by active transport and diffusion. It can then be used by cells for respiration or converted into other substances such as starch to maintain a low sucrose concentration. This increases the water potential of the phloem, causing water to now leave by osmosis. Consequently, the hydrostatic pressure decreases, maintaining the pressure gradient that drives the mass flow. So the movement of the assimilates, the loading, the mass flow, and the unloading in the phloem is known as translocation. Thank you for making it to the end of my video. I hope you found it useful and helpful. And if you have, then please do like it, share it, and comment. And if you're looking for the resources that I've covered in today's lesson, then why not check out my study guide? So that is available on my website, It's available on my website, ukbiotutor.com. So go and check it out. And also, let me know how you found it.